let me give you some background how this all occurred, and also, as you can see in the original slide, Bala is not part of the webinar. So how this all occurred is Hannah Schultz asked in Bala's last webinar, what is the optimal enzyme dose for human islet isolation? And the question really is, it's a nebulous answer because, you know, often people don't know the roots of how this occurred. I wrote a blog post and posted on Monday, which kind of goes, the two pivotal studies were actually done when I was at Boehringer Ingelheim Biochemicals, and we defined the dose for Liberase HI. And the other pivotal study was done at VitaCyte, where Mike Green, who's going to be presenting that data or the design of it, we did a DOE with the University of Minnesota. We paid for the DOE because we had a phase 2 small business innovation research grant. But literally, those are the two studies that defined the dose. I included Bala on the webinar because he did actually all the isolations when we did the design of experiment study at the University of Minnesota. He then also was involved in the Minnesota changing the dose from enzyme, one enzyme bottle to a pancreas to actually doing activity units per gram of tissue. And he did the low study from the low dose from the DOE and did a paper in the American Journal of Transplant. Unfortunately, why he's not here is he's got somebody who's really ill in the family in India, and he had to leave Monday night to go back to India. So he's agreed, though, the next webinar that we will have, he will actually present the data he was going to share on today's webinar. So today, the speakers are myself, who will do the Liberase HI, Mike Green, who was involved in doing the study at VitaCyte in setting up the design of experiment. Mike has also done a lot of islet isolations here at VitaCyte, a lot of porcine islet isolations. And of course, Doug O'Gorman, who's been with, I know Doug was, I think 1998, he's been at the University of Alberta in Edmonton doing islet isolations. And they have been using, what he's going to comment on is over 1,000 islet isolations and doing some analyses and showing what they've found out. And then I'll conclude with a presentation or a summary of what we've learned and how we can go forward. So we'll wait, and this is an open mic sort of situations Teams meeting. So the best would be is to, if you can mute your microphone, and we'll get started. So, getting started. So I'm going to cover setting the enzyme dose with Liberase HI. How this started, it began because, I'll be blunt, it was one of these crazy things. They had extra capacity. This is a European term for didn't have projects for people in Germany, and they had a fermenter, and they decided to make a crude collagenase product, and it was called Collagenase P. And the amazing thing about Collagenase P, it had very high collagenase activity and lower protease activity than what you found in a Sigma type 11, which was, I think, Sigma's attempt at trying to meet the requests at Paul Lacey's lab to try to get a better collagenase. Well, what happened is Camillo Ricordi, who of course was the person who published on the Ricordi procedure and the Ricordi chamber we're all familiar with, he actually ended up finding this product. And basically, and you have to realize this was launched in 1990. He immediately found this product and found out about 30% of these lots were effective in human islet isolation. The biggest problem is that literally the product lost its potency to isolate islets within a year. So what happened? He was continually isolating new lots of collagenase P. So what occurred is that people at Beringer were very concerned, because here we thought this was a research use only product, and find out somebody's isolating islets and putting the islets into people. And so he came out to Indianapolis and basically convinced senior management, because you have to remember Boehringer Mannheim was a diabetes company. It's where they made their revenues. He convinced them they had to contribute to the diabetes community by helping him out to solve this collagenase problem. And this led to 10 people being assigned to do a project. And the challenges of this project was this was a crude collagenase. And I point you to a graph on the left, which was the actually assays reported on a certificate of analysis for all these different lots. You notice that the lots were numbered just from 1 to 45 here. These were the lots that were sent to the US, so that I think they signed their own lot numbers. Lot 9, believe it or not, was the magic lot that Ricordi found, and that's what he came and presented to us. It was lot 9 of this product. But you can see that it has all these lots had very good collagenase activity. The protease activity is misleading because it's azocol. Azocol is a dye-impregnated insoluble bovine hide, which means that it's not only protease it's measuring, it's also measuring gelatinase activity. So this isn't the best measurement for measuring protease, because you get a lot of it, this activity contributed to gelatinase in the collagenase. And clostridium, of course, as we'll see, was a problem. So what Francis Dwellett, co-founder of VitaCyte, did, and he was the guy who did all the protein purification, he looked at all the good lots, and on the right, you'll see the common characteristics of all the good lots. Just to let you know, there's a very nice blog post that Francis published on the whole history of how this product was developed, which, to be frankly, I learned things after he wrote that piece. So if you're interested in more detail, you can go to that reference. So we were able to, believe it or not, with Francis's expertise, really define a formulation within 8 months. And how we tested this is we actually adopted a porcine islet isolation procedure that worked very well with a special group of market weight pigs. And so we knew what the formulation was, and it literally took another year before they could scale up the manufacture of this product. But you can see on the left the comparison between collagenase P and Liberase. These were, again, arbitrarily set. It was a 60-40 class 1, class 2 collagenase ratio, just simply because that was the median value of all the good lots of collagenase. We used 1,650 Wunsch units per of purified collagenase per bottle, 65,000 units. You have to realize we used a caseinase assay. We didn't use this azocol assay to measure neutral protease. And we adjusted the clostridium to 12,000 BAE units. This is an important fact because, in essence, the biggest bugaboo for purifying collagenase is to get rid of clostridium. At VitaCyte, when we started, it took us a year to knock down the clostridium contamination, to minimize it. At the time we launched the product, we had to move, and so in fact, to what Francis's recommendation was, rather than have variable amounts of clostridine, they evened it out 12,000 BAE units. On the right, you'll see what most of you already know. This is an anion exchange protocol with collagenase 2 is the first peak, and you see this, what we know now are degradation products. Collagenase 1 with different degradation products. We know all this from studies we did at VitaCyte. And here you can see on the right how clean that material was for collagenase 2 and collagenase 1. So this was the group that that managed it. I'm here, John Gill, myself, Camilo, and Ray Rajat were really instrumental in getting this project done with us. Terry Federhoff, who I mentioned in the blog post, was really the technical project leader. Francis Dwella, my partner, co-founder at VitaCyte, did the protein purifications, and Sean Lonergan was the next person. Sean Lonergan was the business manager, and Tom Cavanaugh did all of the islet purifications and involved in the islet isolations. So the paper that Federhoff did was published, I think, in '97. It's in the blog post I just wrote. But the interesting thing is what they did. You have to realize we did 26 islet isolations. These were all done at Boehringer Mannheim, where we actually had an islet isolation lab. And what you can see here is that you get almost twofold higher islets with organ donors greater than 25 years compared to those with less than 25 years. Here's the catch, though. In essence, if you put these pre-Ficol EINs are equivalent to islet IEQs per milligram, there's no difference. Again, this highlights the importance, which I've been stressing for quite a little while now, is that we really should be looking at multiple proteases. Proteases are going to be the key, and this will be summarized at the end of the webinar. So the problem that we got into Liberase, as some of you know, was that after Francis and I left the company, there was a switch, and to be frank, the people who were manufacturing the product didn't have the expertise that we knew about in the development, which often happens in industry. And in fact, you start seeing higher amounts of this green bar, this C1b, which indicates that we now know that you're degrading the collagenase. John Lakey did a nice study where Barnett was the first author, who showed that if he used a FTSE labeled gelatin, he calls it collagen degradation units, but it's really gelatinase activity. You see that the unsuccessful isolations on the left had lower gelatinase activity and lower islet yields, where on the right, the successful isolations had higher gelatinase activity and higher islet yields. Gelatinase is a very good surrogate marker for class 1 collagenase, which is consistent with the idea that somehow having these breakdown products wasn't good for isolating islets using the Ricordi method. So I think I'll leave you with this idea that I wrote in a review I wrote on, the evolution of enzyme requirements for a human islet isolation. There were literally 4 different versions of Liberase. The first, as I've already stated, where we added clostridine. The second, where right shortly after we launched the first version, Francis was able to remove the clostridine, and the goal here was to get a cleaner product. At that time, we didn't know the really importance of multiple proteases. We do now. From 2001 to 2003. At the time of the ITN trial, the Immune Tolerance Network trial, which whole goal was to show that could other centers repeat the Edmonton protocol. The group who were involved in that trial modified the dose. And then 2003, somewhere between 2003 to 2008, I think it was earlier, they were unable to replicate the results from our column that was really critical in the separation. And unbeknownst to them, they just were increasing the degradation of class 1 collagenase. I'll leave since, so the whole thing here is that the contributions, I think, were immense from the Liberase project. We developed analytical and exchange methods to evaluate it. What people didn't realize is why collagenase lost activity was because the crude collagenase came pure, it was contained with protease, which damaged it. Once it's purified, it's a very stable enzyme. And no one understood, and we didn't even understand until we did the test, that these products contain endotoxin. We scaled it up. We purified, showed that the purified collagenase protease mixtures could substitute for good lots. We did this very novel approach, which I summarized in the blog post I just wrote, to look at using tissue as a substrate. And in fact, that you really, to do this rapidly, you need to tie the biochemical expertise in testing new formulations with an applications lab. Knowledge gaps, we had no activity assay for class 1. We had no knowledge of structure function. We had no understanding why collagenase P lost its activity, or what were these C1b and C1c peaks that I showed you. We now know the question arose is why were there multiple forms of collagenase? If those who are familiar with the literature, Van Wart defined 7 different forms of collagenase, and we had no understanding of the mechanism. So the last slide I want to show is just something that Bala was going to speak to, and he'll speak to it his next webinar. But problems do occur if you don't adjust the enzyme dose to tissue mass. And this was a paper, as I noted on the right side, by Tatsuya Kin and colleagues from Alberta, and basically looked at 251 islet isolations. They used Liberase HI, or they got collagenase and protease mixtures and could make their own Liberase HI components. Divide up the isolations into quartiles and showed that, in fact, high protease from 0 to 16 to 18 units, or up here at 973 to 2360 NP units per gram pancreas, had suboptimal ideals. And the same thing was for Wunsch. It ranged from 10.8 Wunsch units per gram of tissue up to 81.6 Wunsch units per gram of tissue. So I'll leave you that. And what I'm going to do is turn it over to Mike using a design of experiment approach. I think the important thing here is why we did it is because we knew the history and the limitations of the problem we had with Liberase, and we decided we had to take this into trying to define the variables and understand these variables deeper before we could optimize enzyme mixtures. So Mike, I'll hand it over to you.
Okay, and you're going to advance the slides,
yes.
Bob? Okay.
Yep.
Okay, so in 2010, VitaCyte received a phase 2 SBIR award from the NIDDK to assess the performance of recombinant class 1 and class 2 collagenase for human islet isolation. Now, prior to this time, new enzyme products designed for human islet isolation, such as the Liberase MTF product and VitaCyte's collagenase HA, simply mimicked the Liberase HI formulation that Bob just referred to. And so the formulations weren't a lot different. The pack sizes were all hovering around the 2,000 Wunsch unit target, and they had a mass ratio of C1 to C2 of 60% to 40%. So the limits of the enzyme composition and dose were never really challenged for human islet isolation. So we took this opportunity and used a statistical approach utilizing a DLE to test high and low target doses of recombinant C1 and recombinant C2 in a factorial arrangement of treatments. The doses for the recombinant C1 and the recombinant C2 were targeted as collagen degradation activity for C1 and as Wunsch activity per gram for C2. Shown here on the right are the four treatment groups from our treatment matrix for our 2x2 factorial. And in this case, our high doses, the 20 Wunsch units per gram, were meant to mimic common dosages that were used by leading islet isolations labs. The lower doses then were just 60 and 50% reductions in these targets for both the Wunsch and the CDA activity. Now, in order to achieve these types of targets, we were forced to relax the 60-40 ratio of the C1 to C2 that's in the traditional Liberase and HA products. So that means that at the same time we're testing these four formulations, we're also testing some very divergent masses of C2 and C1 in these formulations. So to our knowledge, this was kind of the first prospective study to utilize a design of experiment approach to define an optimal formulation specifically for isolating human islets. So can you go to the next slide, Bob? So this is just kind of how the DOE was executed. All the recombinant formulations were tested in combination with a fixed dose of neutral protease, and in this case, it's BP protease. And the dose was 24,000 FITC HSA units per gram. And for those of you that are not familiar with that particular assay, that's equivalent to 1.75 DMC units per gram. The formulations were blinded so that those performing the isolations and the subsequent evaluations did not know which formulation they were working with. All isolations were performed at the University of Minnesota using their standardized recording procedure and the co-purification methods. We utilize a split pancreas model for this DOE, such that experimental blends were only tested in the body tail portion of the pancreas. This was then followed by an isolation from the head portion of the same pancreas, which used the standard composition that the University of Minnesota was using, which essentially was a natural collagenase and a CHNP neutral protease at fixed doses of 20 Wunsch units and 1.75 DMC units per gram, respectively. And this was done essentially to serve as a control to assess the quality of the pancreas for islet recovery. All the details and other results from this study have been published in Transplantation Direct in 2016. But for today's presentation, I'd like to share just two slides to kind of lead into Doug's portion of this. So can you go to the next slide, Bob? So this first slide here shows what are called the main effects of recombinant C1. Essentially, it's the differences in the CDA target activity when you go from a low level to a high level. Now, shown on the right are the box and whisker plots for 6 of the measured response variables for this study. And within each plot, the symbols or the little dots, those depict each individual pancreas value. The boxes extend essentially from 25th to the 75th percentile, and the whiskers represent a minimum and maximum range. So as illustrated in the graph on the right, a 50% reduction in CDA target activity had no significant effect on any digestion parameter, whether it's switch time or undigested tissue. Digestions performed with high target CDA activity had comparable islet yields, both pre and post purification, to those obtained with half the CDA target dose, and those are reflected in the center panels there. And lastly, islet function was similar between islets isolated with low or high CDA target activity. So if you go to the next slide, Bob. So this looks very similar to the previous slide, but this is the main effects of recombinant C2 or Wunsch target activity. Again, a 60% reduction in Wunsch target activity yields similar outcomes for every response variable that we measured. So an isolation that utilized 12 versus 20 Wunsch units per gram resulted in comparable digestion parameters, yields, and function. Go to the next slide. So what are the significant findings of this study that relate to identifying optimal doses for isolating human islets? Well, obviously, number one, we did show that recombinantly sourced collagenase is an effective enzyme for isolating human islets. This was also the first study that showed that BP protease was a suitable protease alternative for human islet isolations relative to thermolysin or a CHNP. The surprising findings were that a 60% and 50% reduction in Wunsch and CDA enzyme target activities had no impact on any of the response variables. And this was kind of an unexpected result. Likewise, divergent masses of C1 and C2 were as effective as the traditional 60-40. So we had C1 mass ratios that range from 27 to 55%, and C2 ratios that range from 73 to 45%, and that did not seem to make much of a difference at all in the outcomes of the isolations. So the one thing that this study was designed to do, which failed to do, was actually to define a minimum threshold of enzyme target activity. We really thought we might find a lower limit, but within the constraints of this particular DOE, we did not go low enough to do such. So nevertheless, it did provide us the confidence kind of to move forward with the manufacture of a recombinant blend for human islet isolations. And this formulation is comprised of the center point of this DOE, which translates to 16 Wunsch unit and 150,000 CDA unit targets. So that's kind of how the product was developed. Doug at Edmonton has probably the most extensive experience with this particular product. And I think I'd like to turn it over to him and let him share his experience with using this product over the last several years. So.
Thanks, Mike. good morning, everyone. I'm Doug O'Gorman. I'm the Senior Islet Specialist with the Clinical Islet Lab here in Edmonton. And so, yeah, we're going to switch up the focus a little bit from manufacturing to more application-based. And so when I was approached about joining this webinar and presenting some data, it actually was, the timing was impeccable. I literally had just walked out of an isolation where we had a young donor, and we're trying to figure out what we should be doing, what we should be trying differently to try and, maximize the islet recovery, free islets. And so it really spurred me to kind of go into a little bit of a deep dive within our data set and see what kind of trends and what kind of things I could pull out of our experience. So this is the culmination of that exercise. Just want to emphasize this is largely exploratory. None of this has been peer reviewed, and with the exception of the peri-fusion data, which I previously presented at IPETA in 2023. So if we go to the next slide, Bob, please. Okay, so basically the question that we're asking is, does enzyme dosing based on the pancreas weight drive isolation outcomes? And so I broke that down into 3 different outcomes. We want to look at quantity, so we want to look at the islet yield per gram of pancreas pre-purification as the primary endpoint. We want to look at liberation of the islets from the exocrine, and we want to look at the function. And in this case, we're looking at peri-fusion, GSIRs, and metabolic output that's normalized to the IPN and IEQ. And so this data set that I compiled consisted of just under 1,100 allo isolations. Within this data set, we have 3 different enzyme types. We have the VitaCyte Enzyme, we have Liberase MTF, and we have the RHI products, all with their corresponding neutral proteases, so 2 thermolysins and 1 BP protease. And I went back as far as 2010. It seemed like the appropriate time frame. It allowed us to, it excludes all of the pre-GMP enzymes, but it comes at a point where we saw a relatively stable and plateaued IE yield annually, just based off of our experience using these enzymes. So we can go on to the next slide, Bob. So just before we get into it, just to contextualize and kind of frame my presentation here. Every isolation is a chain reaction. The donor and the process inputs feed the digestion, and we have our outputs, and that tells us how everything went. So before we ask why, we need to establish the vocabulary. And really, with islet isolation, everybody who's on this call who's done even one of them would know that what goes in, quality comes out. And so with the inputs that we have, we're looking at donor age and pancreas weight are fixed by the donor. We have quality and consistency, which are observed by the operators. We have the enzyme concentration and the enzyme delivery quality. And these are really the only 2 things that we have any control of. So out of all the 5 inputs, 2 of them are operator levers that we can pull. And on the opposite side, on the right side, we have our output factors. And these are our readouts as it relates to digestion. Again, we're not necessarily looking at isolation success or transplant conversion. We're looking at digestion success and efficiency. So our output factors for the purpose of this presentation, we're looking at dilution time, switch time, whatever you want to call it. We have our undigested pancreas weight that's remaining in the chamber. We have our tissue weight, and we have a digestion index, which is a composite score that internally we're working on developing. And then we finally have our pre-purification IEQs, which are raw numbers as well as normalized to the pancreas weight. And then finally we have our pre-purification percent trapped. Next slide, Bob, please. So when we talk about dosing, I went back and I looked at our data, and it really isn't, dosing is not on our timeline. It's not a strategy that we have employed from the beginning of, from the beginning of our program, really. You know, we had the same notion as many, where we would throw a full vial of collagenase at an organ, and sometimes if it was a little bit bigger or a little bit more fibrotic, we'd bump it up a little bit. So this is really, dosing is a proxy for product and era up until recently. So in like 2019, approximately, we, through discussions with Bala and Bob, we adopted the RHI low dose protocol and started to target this lower dose. So you can see on this graph here, we get a much tighter band. We have since also, you know, reduced our Liberase MTF approach, and so we do have this low dose protocol which we employ. And so when we're interpreting some of this information, you have to remember that previous experience was there was no intended dose that goes into this, and we had this wide scatter. So that's again just how we're going to frame this moving forward. Next slide, Bob, please. So broke this down into three acts and an interlude in between. Act 1, we're looking at collagenase dose. And so does the dose of collagenase change how many islets we get is the question. Our primary outcome that we're looking at is IEQ per gram of pancreas. And again, this is collagenase focused. I think we have the most amount of understanding about collagenase and its effect on isolation. So this is where we're going to go with Act 1. Next slide, please. Simple scatter plot. We look at, does more collagenase per gram equal more yield? And in fact, in our data set, there's a significant positive correlation with collagenase dose and islet yield per gram of pancreas. However, this is it only explains about 4% of the variance, and at this N of 1100, the finding is trivial and really has minimal effect. So this is a bit of a significant strap here. I don't think there's much to interpret, but wanted to again show this for clarity and transparency. But if you go to the next slide, Bob, when we break this down and we control for organ size, that significance completely disappears. And what we see here is with the low dose collagenase versus high dose collagenase, there's a 75 IE per gram pancreas difference. So there's 1.2% of the mean yield. So again, this is operationally trivial. It is non-significant. And where the raw data showed we had a 351 IE per gram of pancreas gap, this reduces down to 75%. So there's an 80% reduction in that variance between the two groups. So organ size does play a key factor when we're looking at IE per Q or IEQ per gram yields post digestion. Next slide, Bob, please. So after that, we looked at whether the product had any effect on outcomes. And so after correcting for size, this is the same ANCOVA based on with the 3 different collagenase products adjusted for size. There's no difference between the 3 products that we've used. In fact, I think the biggest difference between them is 150 IEQ per gram, 181 IEQ per gram of pancreas, so less than 3% variability. All the CIs overlap on this as well, so there's no product effect. But what will be shown a little bit later is that with the recombinant RHI product, we're getting the same yields at half the dose with a far tighter spread. So our standard deviations are much lower. And this foreshadows a little bit about function that I'll talk about a little bit later. Next slide, Bob, please. When we go in, we look at some of the outputs that travel along with yield through the course of this analysis. This was all done, each number represents a within enzyme correlation based on the yield per gram of pancreas. So there's no product confounding any of these results. And so the three signals that repeat across all three products are digestion time, tissue recovery fraction, and digestion index. Digestion time, there's a negative correlation that's consistent. The longer the digestion goes, the lower the IEQ per gram of pancreas is. The higher the tissue recovery fraction, so the amount of pellet volume or weight that we have relative to the organ size is positive correlation with IEQ per gram of pancreas, as well as the digestion index. This is very logical, makes a lot of sense. The more efficient your digestion is, the more islets you're gonna have pre-purification. Processed organ weight is an interesting one. So the larger the organ, you tend to get more organs, or sorry, more IEQ raw. But as it's when you normalize it to the organ weight itself, it actually goes down. So that's something that you can't control for, but it is an interesting finding. Another interesting finding that we saw was that, again, within the two products that we did not do any dose controlling for, we saw this positive correlation with IEQ with an increasing dose. However, with the RHI, where it was dose controlled, that was completely removed. And so it appears to be just a confounding effect that's spurious, and there really is no dose yield link that we see. So I think this is the cleanest evidence that I can show based on our experience. Bob, next slide, please. So when we put this into a regression model, looking at organ size, digestion time, digestion index, and the dose, collagenase dose, they explain about 18% of the variance. So it's a modest amount, I guess, when we're considering the complexity of an islet isolation from start to end. What this really does demonstrate, though, is that dose is the one predictor that doesn't reach any significance with this. For the other three components, variables, the digestion index, organ weight, and digestion time, all of those were significant. And so, again, this supports the thinking that the dose of collagenase really has no impact on islet yield. But it does also highlight that there's still a very large portion of what's variability that's not explained, and so this can lead to potentially from lot potency, lot variability, obviously donor biology, and operator and process controls as well. So moving on to the next slide, we're going to switch our focus to liberation of islets. So this, again, is more of the job of neutral proteases. And so we're looking now with our outcome measure being, our output being pre-trapped islets and neutral protease dose. So this is a little bit more muddied, not easy to kind of cycle through and pull out the information, but I attempted to do the best here to try and show some application of what we're doing here. So Bob, if you move to the next slide, please. Something that I'm sure everybody, again, is very aware of. Donor age dominates islet liberation. It's a quite linear, quite strong negative correlation. So obviously, the older the donor, the easier, more readily those islets free up from the exocrine. When we look at kind of splitting them into two groups, young donor versus old donor. I know we can draw that line pretty much anywhere along the x-axis here, and you're probably going to see a difference between the two. I picked 45, and it shows here that using 45 as the cutoff point, we see almost a nearly doubled amount of trapped islets in those younger donors. And so this, again, just to frame kind of our approach about what we should be potentially trying, or what we should be looking at for neutral protease dosing, is kind of based off of this scatter plot here. So if you move to the next slide, Bob, please. Some analysis we looked at when we control for age, if we increase the dose of the protease component, do we see an improvement in liberation? And the answer clearly is we do not. Age, again, is dominating this effect, and to a point where we haven't reached that threshold of potentially of where maybe the protease dose needs to be, or the type of protease. But we kept them separate, obviously, because each of them have a different reported activity unit on the C of A. So, and I didn't normalize to a single measure. And it's also there's a nuance to this slide, is that we typically have targeted higher doses at the younger organs. So even those younger than 45, maybe younger than 30, we're trying to see if we can incrementally increase that neutral protease dose to try and free up those islets. But what we have found, at least in our hands anyways, is that we have yet to see any sort of dose effect with young donors and islet liberation from exocrine. Next slide, please, Bob. Again, I think it's fairly well believed that with younger donors, higher BMI, you may see an improvement in freed islets. So I tested that theory with our data set, and it does actually have some merit. Donor BMI versus donor weight, I looked at both of them. Donor BMI was a stronger predictor, so I went with BMI in the model. Fatty infiltrated pancreases potentially have loosened that matrix, and so those islets are more readily able to free up. And so with the younger donor and the BMI, there is a significance. It's small, but in general, what we see is that higher BMI donors, it is significant improvement in islet liberation. But again, donor age is the main driver behind this. Next slide, please, Bob. So kind of just to pull everything back a little bit, when we're looking at digestion, try to introduce a little bit of a QI initiative here that we're undertaking. We're going through our processes and really trying to identify process fails. And so we've done this with various other processes within the isolation. And so, the title here is "Don't ask what dose, ask where it failed." And so I think, again, when we're keeping this enzyme centric within the focus, I think it's very clear, as you'll see, there's a very narrow band of where I think the enzyme actually plays a role in a failed isolation. So I think for the most part, we're doing a pretty good job at utilizing, maximizing our efficiency based on the enzyme we're using, the dose that we're using. If you move to the next slide, please, Bob. So here's my failure tree that we would theoretically sequentially go down if we were to see a failure signal. So a failure signal would be a low pre-purification IEQ, that is expected or unexpected, potentially a high undigested weight and a high percent trapped that is unexpected. So in an older donor, we see a high amount of trapped. We should be going down this pathway of trying to figure out and diagnose where something went wrong. So first, we start with the biological components. Were the inputs adequate for an expected yield, donor age, pancreas weight, cold ischemia time, so on and so forth. If that answer is no, it's likely a pancreas quality issue. It's donor limited and it's outside of our control. If it's yes, we move down to the next. Was the gland fibrotic, soft, or fatty, despite acceptable demographics? If it's yes, potentially there's a pancreas quality issue, although I think, fatty infiltrated pancreas and soft pancreas, it's kind of a maybe. So it could go either way, but again, that's speaking to pancreas quality more than it is anything that's within the operator control. Then we finally get to enzyme. And so now the question is, did we use the correct concentration based on the inputs? And so this is where biology is driving these decisions, young donor versus old donor. This is where I think the biggest block is as it relates to enzyme application. We don't know what to do with young donors as it relates to NP dosing, or NP type for that matter. So we have to look at that, and we have to go back and we have to review these cases and see what differentiates the successful versus the non-successful in those cases, and see if we can optimize and refine our processes for that. But it does, again, enzyme represents a pretty narrow band of failure. If enzyme, though, is deemed to be appropriate, we look at enzyme delivery. So the cannulation, was cannulation successful? Was perfusion successful? Yes, we move on. No, there's a process error. And then finally, digestion assessment. Again, was it over-digested? Was it under-digested? And these would speak more to a human process error. And so I think this is a good strategy, and it's a good control to put into processing, as it allows us to frame each isolation, as you'll see on the next slide, or in a couple slides, where these land will really tell the story. So, nope, that's okay, Bob, you can go. Yeah. So the digestion index, which I referred to earlier, that I included in one of my models for analysis, is basically a combination of two terms. We have the fraction of digested pancreas, and we have the pellet recovery on the other side. In this equation, we have the fraction digested being the organ weight minus the undigested weight divided by the organ weight. So that's the percentage of that whole organ that you've digested. And then on the pellet side, we have the pellet weight divided by the organ weight. Very simple. I have them weighted equally, just again, because this is more exploratory and definitely would need to be optimized, but each term is weighted equally right now. That could be altered, where the pellet recovery is weighted a little bit heavier than the fraction digested, based on the fact that we do see this positive correlation with improved IEQ per gram of pancreas with pellet recovery. So when we work through some examples here, we have each row would represent a different isolation, different digestion, where we have an organ weight of a standard size of, for example, 85 grams, and we have a good digestion where we have a good pellet weight, we have a very low undigested weight, and so we have a digestion index of 80.6. Next one, we have a high undigested organ where the pellet weight is quite low and the undigested weight is quite large in the chamber. This would maybe speak to organ quality or potentially enzyme delivery. Then we have a low pellet recovery where, if you go across, you can see that despite the organ being 85 grams, we only recovered 18 grams, and there was not much tissue remaining in that chamber. So was this over digested? Was it fatty infiltrated? Is there organ viability concern that's causing the cells to just dissipate into single cell? And then we have like a true failure where everything is bad. And that one's a little bit harder to diagnose potentially what the root cause is, but this is kind of the approach that we're taking when we're assessing these. And so when you apply this to our data set on the next slide, Bob, you can kind of see where everything lands on this scale. And we broke it down into quadrants here. And again, where an individual isolation lands, that's going to be its signal, its failure signature. And just to clarify this graph, because there's a lot going on with it, age is differentiated by color. So the blue would be the under 45 group, and the purple is the older 45 group. And then the size of the bubble is the percent trapped that we calculate. So as you can see, we don't really have an issue digesting young donors' pancreases. We tend to completely digest them, and we tend to recover almost the entire amount of that organ. What we see in the older donors is we typically have a lower recovery fraction, even if we digest the entire organ. And then below the y-axis, there's 70%. That's where we're getting into maybe there are some issues. So again, this is just something, a tool that can be used to really evaluate and categorize how efficient our digestions are based on the enzymes that we're using at the doses that we're using them. So just a little bit of some insight into some quality initiatives that we can apply to enzyme digestion or pancreas digestion and enzyme deployment. Next slide, please, Bob. So the final question that we had was, does dose affect function? And here we actually find where dose matters and lower is better. We're looking at perifusion of transplanted islet preps, and we're looking at the GSAR, we're looking at metabolic function. So this was presented in 2023 in IPETA. Next slide, please, Bob. And I'll just quickly summarize this, but basically what we found was that when using a low dose RHI product, we saw significantly higher stimulated insulin secretion in response to glucose. And so this, when we looked at this, it carried through all of our different metrics. When we looked at peak glucose or peak insulin secretion, it was higher with the low dose. When we looked at area under the curve on the perifusion, it was significantly higher in the low dose RHI. When we looked at metabolic output and rates, so these are measures that are normalized to the amount of the IPN or the IEQ in that individual chamber. These are also significantly higher in the low dose group. And so this is just a quick one slide to show you that we've established that the low dose has some improvement on function. My theory is maybe that the ECM is not being disrupted as much with the low dose, or perhaps it is a product related phenomenon. We did test the low dose MTF protocol with this, and we did see a higher stimulated response as well, but at the time the MTF arm was only 5, an N of 5, so as opposed to an N of 30 and 90, so a much lower number. So they didn't show any significance. I have not gone back to rerun this data, but maybe it's okay to assume that a low dose, regardless of the product, would be beneficial to the islet function. So just a couple more slides. Next one, please, Bob. So when we kind of circle back to the beginning, does islet, or sorry, does enzyme dose control outcomes in an islet isolation? The three questions we asked: does it change the yield? No. Collagenase dose nor product have any effect on the yield, at least in our experience. Execution, digestion time, and digestion index, that's what's predicting the yield. Second question: does neutral protease dose free more islets, increase neutral protease dose free more islets? The answer is no, at least in our hands. Neutral protease dose is dull across all three proteases. Donor age and BMI are driving the liberation of those islets, not the neutral protease dose. And then finally, third question: does collagenase dose affect quality? And in this case, yes, it does. Lower dose islets show significantly better GSIR and metabolic output. So lower is better. And so if I can leave you with one message, again, based off of our experience, it is that standardizing to a lower dose protocol costs no yield, does not impair liberation, and improves islet function. So this is a clear quality win. And I think it really represents maybe, and it's crazy to be saying this after 25 years of doing this, maybe this is where the starting line is for really doing an effective enzyme dosing study on a large scale. If you go to the next slide, Bob. So that's where we have to go to next. So there's some still open questions that I wasn't able to answer today. They span across a couple of different areas for sure, but just briefly to look at them, we have enzyme lot potency, so batch to batch variation within the single product. This has not been tested by us here. I suspect that there's not much anecdotally, but we would have to go back and do that. More importantly, I think, as it relates to enzyme lot potency, is we all know that the one-shot assay is not a good primary dosing metric. It's fundamentally flawed. It's used because it's a more stable measurement that they can put on the C of A. So the development of the CDA assay and the CDA units and the dosing of that needs to be looked at more in depth, and that'll be more precise and more specific, and may contribute more to this story. Second is donor biology. We show that age and BMI are explaining a large portion of islet liberation. Did not factor in cold ischemic time in this. Cause of death, the donor type, DCD versus DBD versus ANRP versus MAID. Like there's a number of different components to that may be contributing, that's underlying. The percent trapped interactions, all that, we need to push that forward. The only user control that we have for this is through donor selection. It's generally our practice that we don't necessarily select donors based on the potential outcome. We have a young donor. We know that it likely is not going to proceed to transplant, but that does not impair our efforts or deter us from accepting and trying it, because occasionally they do work. We have operator effects. Again, this has not been modeled yet, but there's execution variables that are very critical to outcomes, and digestion time, digestion index are what's driving the yield here. So who and how the isolations are being run probably matter. Experience probably does matter in this. Digestion, trying to train somebody on how to assess a pancreas digestion is one of the most challenging things that I find personally, with new hires, with new staff. It requires a lot of experience. And so, even a couple of extra minutes may have significant negative effect on the outcome. So again, that does contribute to it. So can digestion time be altered via some sort of optimized process control, whether that be AI or something beyond that? I don't know, but maybe there is an opportunity there to optimize. And then finally, prospective dose titration. This is going to be the causal test, randomizing or matching low to high dose within one product and a narrow donor age band. This is really what's going to be critical for the next steps, specifically as it relates to neutral protease dosing and freeing up islets. I think that's really what needs to happen in order for us to find some real success with young donors. In Canada, specifically, we've seen a fairly significant decrease in donor age over the last number of years because of the opioid crisis. And so our donors are becoming younger and younger, and they're becoming more challenging to isolate and convert into transplants. And so this is something that really does need to happen. But it is challenging to do it in a regulated clinical setting. So there needs to be some controls as well for that. So I'll leave you with that final question. Again, we come back to, is there an optimal dose for enzyme? I'm not sure. I probably could have saved you about a half an hour, 40 minutes if I opened with that. But I think that we're getting closer, and I think we have a better, we're in a better position to frame that question and actually answer that question moving forward. So thanks for your time, everyone, and I look forward to the discussion after, and I'll hand it back to you, Bob.
Yep. Thank you, Doug, and thank you, Mike, for doing this. I'm just going to end up with some closing comments. I basically said that what we didn't know in knowledge gaps, I think, as you can see on this slide, we have answered them in 2026. And since time is late, I'm not going to go through them. But the bottom line, from a biochemistry standpoint and a manufacturing standpoint, I think we've answered the collagenase problem. What is Doug nicely had summarized, there's still a lot we've got to learn about actually taking this knowledge. And as I said before, and you'll see in a slide shortly on the mechanism of action, it's the protease that's got to be worked on. So getting to the next slide is just some slides lead up to a 2-minute video. You have to realize that the extracellular matrix is a jungle of macromolecules where collagen, fibrils, fibers predominate, and they're literally entrapped. And what happens, and people who've done this in the past, is that actually just adding neutral protease to the tissue doesn't digest the tissue, and that's because collagen provides a protease-resistant superstructure. And basically you have to have the collagenase, which not only cleans out, if you will, or thins out the collagen fibril jungle, but it also loosens up the matrix. So now that these anchoring proteins that either directly or indirectly hold the cells to the extracellular matrix are now being able to get cut. So we've been working with Josh Saccone. There's only 2 people in the world who actually are studying this mechanism of action. And he's been very helpful to us to try to make sense of how the collagenase works. And this is a slide from his lab. And here they're using recombinant collagenase. Collagenase G is class 1. Col G is class 1. Col H is class 2. In class 1, you think about class 1 as a plane shaving off a tropocollagen molecule at a time, whereas the class 2 is a saw. It breaks it apart. And the beauty of this, you have to realize, is that's the unique thing about bacterial collagenases. They have to break down the collagen so they can eat. So they have a broad spectrum against over, excuse me, all different classes. I think the interesting thing, and I mentioned this in the blog post I published earlier this week, is it was a very unique aspect of the Liberase project was to use the tissue as a substrate. And in fact, you can see that as you measure just the complete digestion of a porcine pancreas, you can monitor the islet release and islet size. And you do see two phases of islet release, small islets at about 16 minutes and then a maximum release at about 37 minutes. Just to show you that, you know, it's a complicated process. And I think at the time when Liberase started, you just stop the reaction. Of course, now you continue on and keep on harvesting islets. So you are seeing I'm sure, this two-phase release. What I'm going to show you now is a video. It's only be 2 minutes of medical animation we did, which I think clarifies what's going on. So I'll end with this last slide. The way can move the field forward in improving islet yields is there has to be an industry end user collaboration, and the field is stuck because as mentioned before, that Doug is the problem with young donors. We have to increase islet yields and also increase islet yields in chronic pancreatitis patients undergoing TPIUT. I think to move the field forward, you have to understand how to control the reagents, the enzymes used in these processes. And we started using several years ago we use of Purified defined enzymes. And it was really because we showed that with Minnesota that intact class 1 collagenase was really critical to increase islet yields using the recording method. This is aligned with an ISO 20399 change control. The ISO 20399 is the standard for ancillary materials that was published in 2022. And the question is you really need to know the impact of the enzyme characteristics on the application results. And just to reiterate, we're always open to establish scientific collaborations to address these challenges. And I leave you with this note from the ancillary materials standard in section 5.2, which basically says if we're going to move the field forward, both parties, the ancillary material user and the ancillary material supplier, have to have a cooperative and transparent working relationship. Otherwise, we're not benefiting. And this is really important with technical support from our side. It's really, we would like to understand what specifications you need for your isolations, for any cell isolation. I will mention this at this time, we actually do have a colorimetric collagen degradation activity assay that's a transferable assay. So we're hoping to get this out in the near future. We plan to publish a tube-based protocol on protocols.io, and the part of the kit-based protocol will be a microtiter plate format. So with that, we are ready to answer any questions from the audience. And I appreciate your patience in staying over the hour to hear all of this webinar. Any questions? You can unmute yourselves and just ask.
Yeah, hi Bob, thanks. This is Greg Zott. Can you hear me?
Yes, I can hear you.
Great. Thanks, Doug and Mike, for those great talks. Really informative. I have one question to Doug. You know, you said the size of the organ may be an issue. So if you get over 85 grams, do you feel the limiting factor in that is maybe the chamber size and the volume of solutions that we're using during the digest?
I had not considered that. I mean, I don't think we have come across a scenario where the chamber size and the volume has been an issue. In fact, I've often wondered maybe we need to increase the concentration of the enzyme in a smaller volume when we're perfusing it. I don't know what the outcome of that would be, but no, I had not considered that. I don't know exactly how that might factor into it, though. I don't know if you have any thoughts, Greg.
Well, no, I kind of like to do the smaller volumes, higher concentrations, and then the standard 600 ml chamber. And yeah, I don't know if going larger or adding more solution would actually help on larger pancreas, especially pancreas maybe over 95 or 100 grams. But yeah, I was just when you were giving your presentation, that's something that just kind of popped up. So I was wondering if that was a limiting factor when you get to larger pancreases. What could that be in your yields going down? So
I don't think it's a limiting factor.
I thought you said.
I mean,
I'm sorry.
No, I think what I was referring to, as the organ size increases, actually the IEQ per gram of pancreas goes down, not necessarily the completeness of the digestion.
Okay. Oh, then
Sorry, I might have misunderstood that.
Yeah, I may have misunderstood you too. Okay, thanks.
Hello.
Hello.
Sorry. You can go ahead if you want.
Yes, this is Bashu Naziruddin. I'm calling from Dallas, Baylor. I have a question for Doug. Thank you for a very good presentation and the summary. During your experience, the islet size, when you get large size islets, when you get moderate size islets, you get small size islets, have you analyzed that, and does it play a role?
That's a good question. I have not directly as it relates to enzyme deployment. Certainly, we've looked at islet size and distribution of islets as it relates to culture recovery and potentially factoring that into, the functional dose of an islet going into the recipient, because those islets you would tend to lose those in the peritransplant, but not within the digestion. But it is an interesting idea that, that's something that probably we should go back and take a look, because I mean that's the starting point of with those islets potentially, where that hypoxic environment is the first being introduced to those islets in a release fashion. So yeah, it's something that we should maybe go back and take a look at. It's a good point, Bashu.
Okay, I just have a question. I actually mentioned it in the chats, but I had to learn every case is different. So how do we go about dosing if let's say we have two pancreases about the same size, but they end up with different IEQ yields?
So I think as it relates to the dosing strategy, I mean, if they're comparable donors, age, weight, BMI, cholestemia time, all those, you would probably be using the same dose for both. With as it relates to the IEQs that you're recovering, that's where I think you need to kind of go down through that failure tree and see if you can identify if there was some underlying biological component that's driving that, or was there an issue with the process and in terms of digestion assessment or perfusion or cannulation, and see if you can or maybe it's a combination of a couple of them. But I think as far as the strategy goes, you would apply the same strategy across the board, and then if it does fail, if there is a process failure, that's where you're going back and you're trying to resolve that down to maybe one or two potential reasons.
Okay, thank you.
Yeah, Mike, Greg Zott asked a question in the chat. Maybe you can answer this. Is in this study, is there a standard calcium level that was used in both the 50 and 60% reduction groups?
I would have to check on that, but I'm pretty sure they just use the routine HBSS buffers that they normally use. I don't think there were any adjustments to the calcium levels.
Yeah, I don't think that's an interesting thing, and I don't remember the specifics because there was a paper by Korsgun's group that showed that maybe you need extra calcium, I think, in the loading, but maybe Doug, you know something about this. But from a biochemistry standpoint, it isn't for the collagenase, so I'm not sure, where this fits. So.
Oh, yeah, I could comment. This is Greg.
Yeah.
No, I think it was Daniel Brainhorst that was responsible for some of that. And
Okay.
I know I've increased my calcium over the standard HBSS levels to get a little better activity. So I think I'm at 35. It's a millimolar calcium. I got to go back and check, but yeah. And we found that, especially when we switched to the Nordmark enzymes back in 07, or we definitely needed more calcium in the reaction to get improved digestion.
Really?
Yeah.
One other comment, just to end up, because we're about coming to the end of the event. I think the interesting thing that we learned about the model that we have shown you in the medical animation is the fact that if you have a really good clean collagenase that isn't contaminated with other proteases, you can now start to dissect what's really important. And Nazir gave a very nice earlier webinar on the importance. What he found was one of the forms of laminin actually correlated with lower islet yields in TPIAT. And I think this again begs the question, and that we have to explore the use of complementary proteases. And this is again consistent with the original report by Terry Federhoff, which shows that when they had the clostripane, high levels of clostripane together with the collagenase, and at that time thermolysin, you got really no effect of freeing islets from younger donors. So it's one of those things that the biggest problem the field has right now is that there's not a lot of money to go around to look at these really process improvement questions. And it's something that, as I said in my concluding slide, we're more than willing to work with people to start exploring scientific issues to do this, because I think we all benefit.
Yeah, it's Greg again. I think that's a great comment, Bob. However, I think when we've in the US, we've been stuck kind of doing more autologous organs, and we don't have the luxury of doing some of the experimental on highly fibrotic, diseased pancreas.
That's
we don't
right.
have that luxury. So, whatever information we could glean from the cadaver donor, it will benefit the auto islet processing, and maybe hopefully answer some of those questions, which I think you and I tried to do several years back, but we never really concluded it.
Never really panned out. Yep.
Yeah, but we have the intact C1 versus C2 concentrations or ratios, and I think that might play that having that information might be helpful with the auto islet programs. Yep.
No, I agree. But yeah, we're at a point where I think the field has shown definitely where we need to go. And again, I thank my Mike to show everyone what was involved and why we did the study the way we did it, and Doug, of course, to give always an excellent summary of what they've learned from Edmonton, because they've done so many more isolations. So it's, I think, a great webinar. I'll leave it that what we'll do is Bala will, once he gets back from India, we'll set a date for a follow-up webinar, which will complement this webinar in discussing what he's found about islet dose. So again, I thank you very much for all your time, and the session will be recorded. And so feel free to pass on, if you enjoyed it, pass on the link to other people, other colleagues. Thank you.